Welcome back. Today I'm going to talk about B cells and antibodies. The immune system depends on multiple lines of defense when it encounters a pathogen. One of the key lines of defense is the development of humoral immunity, which is the secretion of specific protective antibodies over long periods of time. What are antibodies? Antibodies are proteins produced and secreted by B cells. They bind foreign substances that invade the body, such as pathogens. Antibodies also are an informative biomarker for previous exposure to a particular pathogen. So an antibody is a Y-shaped molecule that has a constant region and a variable region. And it's this variable region that's really important. So this variable region can recognize antigens, millions and millions of different kinds of antigens. So we have antibodies in our body that are specific to multiple different pathogens and immunogens. What types of antibodies do we have? We have IgG antibodies, IgM, IgA, IgD, and IgE antibodies. IgM antibodies are the first antibody produced upon antigen invasion. It's also the first antibody produced by the fetus. It's efficient in binding antigens or pathogens that have many repeating structures, and an example of that is a virus. IgG is the most dominant type of antibody that we have in our immune system. 80% of all antibodies are IgG. This antibody is critical against virus infections, bacterial toxins, etc. These IgG antibodies can be classified into 4 subclasses: IgG 1, 2, 3, and 4. IgA plays an abundant role, and it's the most abundant antibody found in tears, saliva, breast milk, and in mucosal tissue. So if you have a respiratory infection like the one that we are interested in, which is SARS coronavirus 2, you will generate a lot of IgA antibodies, and you might find IgA antibodies in the respiratory tissue. The other isotypes of antibodies, such as IgG, M, E, and D, can be found mostly in the circulation. So we have thousands of lymph nodes in our body. Lymph nodes are these small bean-shaped structures present in all parts of our body. So within these lymph nodes, we have T cells and B cells that are waiting to see if a pathogen comes by. So if a pathogen such as a virus infects a human, this virus then travels to the local lymph node. The lymph node, which has T cells and B cells, then get activated. The T cells get activated by B cells presenting chopped parts of the virus, and the B cell then gets activated in turn after interacting with the T cell. So the B cell then becomes activated and becomes this antibody secreting factor known as a plasmablast. So these are giant B cells that are highly activated, that are pumping out a lot of antibodies. And these antibodies are critical in preventing us from getting more sick. A subset of cells go on to become long-term memory B cells. So these long-term memory B cells, all plasma cells, are highly critical for us to maintain long-term immunity. These plasma cells secrete small amounts of antibody. They're present in the bone marrow, and they constantly secrete antibody, feeding the circulatory system. So in our bloodstream, we have antibodies specific to a pathogen. So for example, in people who've been exposed to SARS coronavirus, the virus comes in, activates T and B cells, then B cells that are specific to SARS coronavirus start secreting antibodies. So you get antibodies that are specific to that virus. The antibodies circulate in the blood. Some of the B cells, hopefully, some of the B cells become memory B cells. And we also have the generation of long-lived plasma cells that are SARS specific. And these plasma cells will then feed antibodies into the circulation. So in the circulation, several months after we've had an infection, we should have antibodies specific to the SARS coronavirus 2. In addition, we will also have antibodies to everything that we've seen before. So most of us have been exposed to influenza. most of us have had vaccines. So we'll have antibodies to measles, mumps, rubella, influenza, rhinoviruses that cause the common cold. So the bloodstream is full of antibodies specific to multiple viruses, etc. So it's complicated, and there's all types of antibodies that are present that are specific to these pathogens. The hope is you generate long-lasting protective antibodies. So shown in this example are days after infection versus antibody titers. So if you get infected with pathogen A, in this case, pathogen A is SARS coronavirus 2, you will start generating antibodies, both IgM and IgG antibodies, few days after you've been infected. So the IgM antibodies are the first antibodies to come up, and then you have IgG antibodies coming up after IgM antibodies go down. So you have the generation of IgG antibodies. They last for a certain period of time, and then they decline. So what happens when you are exposed again for the second time to the same pathogen? So 6 months from now, if you get exposed to SARS coronavirus 2 again, is that going to be helpful? So when you get exposed again, what happens? Your memory B cells and your plasma B cells start secreting antibodies much sooner. Now you have the rise of antibodies in response to the second exposure. So this is the second time you've been exposed, and these antibodies rise to a much higher titer compared to, say, if you've had an infection with a different pathogen. So pathogen B, for example, could be a rhinovirus. And a rhinovirus is not that similar to a coronavirus. If you had a second infection with a completely different pathogen, these kinetics would be similar to what you had during a first infection. But if you have a second infection with the exact same pathogen, so pathogen A, you will have a much higher increase in antibody titers compared to the first infection. So that's the advantage of having immunological memory. So in the previous slide, it was hypothetical data should you get exposed to SARS coronavirus 2. In this slide, this is taken from a New England Journal of Medicine article from 2003, in which patients who had developed SARS infection, so this is not SARS coronavirus 2, but SARS, they had collected serum 1 week after people got sick up to 12 weeks. So they had multiple samples taken from patients, from about 20 patients who were infected with SARS. They also had 103 healthy contact controls. So these are the negative controls in their study. So what they did was they developed assays to detect antibodies to specific proteins of the SARS virus. And this is looking at actual data. These are weeks after symptoms, and these are the antibody titers, both IgM antibody titers and IgG antibody titers. So they follow typical kinetics that we expect with most viral infections. So IgM responses in these 20 infected patients were the first antibodies to rise. They were detected in the serum for several weeks, and then they decline. This is what you typically expect. IgM antibodies last for a few weeks, and then they decline. IgG antibodies in these 20 patients took a little longer to start to be detected. They kept increasing, and then they lasted for several weeks. So the data goes until 13 weeks after infection. So the IgG antibodies to SARS coronavirus seem to be long-lasting. The latest time point that they tested was 13 weeks. So that's quite encouraging, and this is to SARS virus from 2003. As I've mentioned before, SARS virus from 2003 is quite similar to the SARS coronavirus from 2019. So the question really is, do we detect antibodies, and how long do they last in patients who've been exposed to SARS coronavirus 2? So we've only known about SARS coronavirus for 3 months. However, there's a paper out this week where they've looked at antibodies that are specific to SARS coronavirus 2. It's the first report and very preliminary. There will be several articles that come out in the next few months that will be much better powered, will include several more patients. This is a study of 34 hospitalized patients who were admitted in February of 2020 with confirmed SARS coronavirus 2. And as suspected patients who were exposed to SARS coronavirus 2 developed IgM antibody responses. Those IgM antibody responses were the first to rise, and then they declined, which is what you expect to see. IgG responses were also detected in many of these patients, in most of these patients, and were long lasting. But this is only up to 6 or 7 weeks. I mean, it's just been, they were admitted in February, so it's amazing to me that there's data already published, but there's more to come. So I'd like to end this talk by telling you that there's a lot to study about B cells and antibodies. I gave you a very brief introduction to B cells, the types of antibodies, how you generate an immune response, what's a primary response, and what happens when you likely get re-exposed to the same virus, and some data, some primary data looking at antibodies to both SARS virus that was an epidemic in 2002, 2003, and SARS coronavirus 2, which is the current pandemic that we are experiencing. So hopefully next time I will go into some more details about how you look at long-term immunity. How are you better protected if you have a mild infection? What kinds of antibodies do you expect? What's the function of the antibody that you should look for? Thank you again for your attention. This is Anuja Matthew, and hope you enjoyed this lecture on B cells and antibodies.